I'm Preethika Kankanala, and today I'll be talking about the principles of genetics. So we'll go over heredity and variation and how variation plays a role in evolution and how some traits resemble the parents, the traits of the offspring, and how some variations are important to be different from the parents so that like it helps in evolution and like better survival and better adaptation to the environmental factors. Um, so these are the objectives of the presentation. We'll go over the basic concepts of genetics and a little bit about the father of genetics, Gregor Mendel, and the concepts of genetics that was discovered by Mendel, and the three basic principles that plays a role in the basics of genetics. Then the different branches of genetics, we won't be talking much about that because that's too complex. Then the basic terminologies in genetics that we use, and a little bit about the pedigree, just the introduction, the definition, and the whole concept of pedigree, why we use it in medicine, and the symbols that are used in pedigree. So now what's heredity and what's variation? So just remember, heredity is resemblance, variation is the variation. So that's pretty easy to remember, right? So heredity is the tendency of the offspring to resemble the parents. So we inherit like one half of genes from both the parents. So we resemble our parents a bit, but there's also a huge degree of variation, and this variation depends on like mutations that happen during the inheritance, and these need not be like pathological mutations. It's just physiological mutations that happen so that we are better fit for survival, better fit to the like environmental conditions. Okay, so this is Gregor Mendel. He was an Austrian monk, and he did most of his practice in Czech Republic. He's known as the father of modern genetics, and he derived the principles of genetics based on just one experiment. And this was an experiment in pea plant where he studied the law of dominance and law of segregation and law of independent assortment, all by looking at the physical characteristics of the peas. So the round and yellow peas, and then the wrinkled and the green peas, where he did a hybrid cross of the peas, and then he derived these basic laws of genetics. So these are some terminologies that we use. So the term genetic was coined by William Bateson, but the terms that we use now, gene and allele. So gene is the hereditary determining factor. So when we talk about gene, we could be talking about the eye color or the hair color or height. So that is gene, where you're talking about the heredity determining factor. And it's a segment of DNA that's located in specific position. So you have the locus here. But allele is the variation. So think of gene as the hereditary factor and allele as the variation. So when you talk about eye color, that's a gene, and we get the genes from both our parents. But the allele is it's the alternative or it's the variation. So when we talk about blue color of the eyes or brown color of the eyes, that's the allele that we're referring to. And the alleles can be dominant alleles or recessive alleles. So the dominant allele, it always expresses itself. So when we talk about height, we have the dominant trait, which is the uppercase T, and then the recessive one, which is the lowercase, as you can see here. So two characters, tall and short, and the dominant one is uppercase T, and then the short character is lowercase t. So the dominant allele always expresses itself. This means that you could have both the dominant traits inherited from both your parents, and the dominant allele is uppercase T, so you express that. But even if you have a heterozygous allele, which means you have one uppercase and one lowercase, you still express the dominant allele. But for a recessive allele to be expressed, you need to have both the recessive traits. Yeah, then we have genotype and phenotype. So as we can see here, we have TT, TT, and PT. So I highlighted this in blue. In this case, the genotype is different. So you can see that this is homozygous, which means you have the same trait, and then heterozygous, which is different. But the phenotype is the same here because the dominant the dominant allele always expresses itself. But for a recessive one to be expressed, you need to have both the recessive traits. So the genotype is the genetic makeup. As you can see here, you have the different genetic makeup. But phenotype is the physical feature or the appearance of the organism. So when we talk about genotype, it's like both capital T's or like one capital and one lowercase. But phenotype is the physical feature, which just means tall or short, nothing else. So phenotype is the expression of genotype, but phenotype also has some environmental factors that play a role. So genotype is just what you inherit from your parents, but for phenotype, it's an influence of both genotype and environmental factors. So even if like both the parents are of medium height or short, the offspring can be tall. And this is when like environmental factors play a role. So they could be like good at sports or like regularly exercise. So they might be taller than their parents. So the phenotype has has both genetic and environmental influence. Then we have homozygous, heterozygous, and hemizygous. Homozygous is when the allelic pair are identical, as you can see here, like purely dominant and purely recessive. Heterozygous is when the allelic pair are not identical. So you have one dominant and one recessive. Hemizygous is when genes are present in only one copy. So in men, we like men have XY chromosomes, right? So you have total of 46 and 44 plus X and Y. So this is only one copy. So the genes are present in only one copy. What's present in X is not present in Y. So it's only present in one copy. But for women, it's XX. So you have both X chromosomes. Then we have hybridization and hybrid. So it's the process of two genetically different individuals. So try to relate it to the words that you hear in real life. Like when you talk about a hybrid dog or a hybrid fruit. So when we do hybridization and get a hybrid product, we're focusing on the traits that we are interested in, that we want, and then we make a hybrid cross and get the desired product. So monohybrid cross is when you just take one trait, so tall or short. Dihybrid cross was the experiment that was performed by Gregor Mendel, where you have two traits. So you're looking at the color, yellow and green, and also the characteristic of the pea, so round and wrinkled. So this is the parent generation, represented as P generation, and law of dominance. So you're only taking one trait, and when you do a cross, you get the first generation. And then the law of segregation, like we'll go over it soon, because you can see in this slide. So first he took round and yellow seeds. For the P to be round, it has to have the uppercase R, right? And then for it to be yellow, the uppercase yellow. And then if both are recessive, then you would get wrinkled and green seeds. So we did a dihybrid cross. So for the first generation, as you can see, so this one is purely round and yellow, and this one's purely round and green. So we can just take one R and one Y here, and one R and one Y here. And we would get the first generation cross, which is one uppercase R, one lowercase r, then one uppercase Y and one small case y. And then we split it into four combinations. So just take the first letter of each. So R and Y, so both dominant here. And then lowercase r and uppercase Y, then uppercase R, lowercase y, and then both lowercase. And then we make a cross. So you represent all these gametes on like let's just call this y and x axis. Okay, so we represent all the gametes here and here, and then this takes some time. This would take up to like 2 minutes to draw the whole thing, and then he analyzed the traits. So he got when he just looked at yellow and green, the yellow piece after cross, um, he again segregated that into round and wrinkle. So out of the yellow piece that he got from the dihybrid cross, how many were round and how many were wrinkle? And he got the ratio of 9:3. And then again, the green ones, how many were round and how many were wrinkle? So this is law of independent assortment, where the alleles for separate traits are passed independently of one another. So this means that you don't always get yellow seeds with round seeds. It can be round and green. It can be wrinkled and yellow. So this is law of independent assortment, where the traits are passed down separately and they're independent of one another. And the previous one that we looked at, like tall and short, that would be monohybrid cross. And then law of dominance is different, where the dominant trait is always expressed, But for recessive to be expressed, you need to have both the recessive alleles. And this is a Punnett square. So you can see here. Let's say that we have a heterozygous dominant trait, so capital A and small a. You just draw a Punnett square and then you get the possibilities, the genetic possibilities. You can also do this with blood groups. Like if you know the blood group of both your parents, then you can just draw it and see the possibilities. Then this is a pedigree. It is a graphical representation of like of various generations, and like you should also show how they're genetically related to each other, the offsprings, and who is affected in the family with a certain disease, and why we like, and they would be the index case. And then we try to analyze like who is a carrier and who is affected and who is not affected. So each member is represented by a square or a circle. So if it's a male, it's a square, and then if it's a female, it's a circle. And they're connected to each other by relationship lines. So as you can see here, the first generation, there is one horizontal line, which means that they're married. And then the vertical line means we're talking about the offsprings. The proband is the member through whom a family with genetic disorder is first brought to attention. So let's say that I am a proband and I'm affected by a disease. So I'm trying to analyze the entire like family. So I'm trying to depict the entire family through me. So I am the proband. So the symbols used, we've gone through this. Then if the sex is unspecified, if you're not sure, like let's say 2 generations ago, you know that like there was another child like born to your grandparents, but then you don't know the gender. So if the sex is unspecified, then we use the diamond symbol. And number of children of sex indicated. So this would just be like, yeah, the numbers. If there are like too many kids and you don't have to look into each one of them. So if you're affected by the disease, so if someone's affected by the disease, then you draw like obviously the circle or the square. And then if it's like fully colored, then they're affected by the disease. And heterozygotes for autosomal trait. So if they're a carrier, then you represent it like this. Then carrier of X-linked recessive trait. So for X-linked disease, um, yeah, so for an X-linked disease, if it's a recessive trait, then the woman is not affected because for a recessive trait to be expressed, both of them need to be a carrier. But for a man, even if he carries the X-linked, the affected the X chromosome, he would be affected with the disease. And we went over the proband already. So this is the prenatal death, and then miscarriage is represented like this. Then adopted into a family, adopted out of a family. Then this is marriage and this is extramarital mating. Then consanguineous marriage and divorce. Monozygotic twins, dizygotic twins. So this is an example of a family pedigree of an autosomal dominant disease. Because in autosomal dominant, you would see the disease in like if you see the disease in every generation, then it's autosomal dominant. And this is so when a new mutation arises and you're trying to analyze where they would have inherited it from. So as you can see, so this is very obvious that it would be a autosomal recessive disease. Then this is an example of a pedigree of X-linked disease. So as you can see, the mother could be a carrier or maybe not, but she's not because we didn't have the round here, like the concentric circles. So the father is affected with the X-linked disease in the first generation. And then in second generation, you can see that both the females are affected. So for a female to be affected, we know that they need to have both the X chromosomes. So the mother is a carrier in this case. Then in third generation, again, you can see that like the male is affected and then again another female. So an advice tips for pedigree, always draw male partner to the left and each generation should be in the same horizontal plane. Maintain that because if you don't maintain it in the same horizontal plane, you might think that the first generation belongs to the second generation and it gets chaotic. And always, like whenever possible, draw siblings in birth order. And if that's not possible, record the age. Then each generation can be identified by Roman numerical. So as you can see here, And what's the use of pedigree drawing? To establish mode of inheritance and to know mode of inheritance if you don't know if it's autosomal dominant, autosomal recessive, or X-linked disease. So you identify the individual at risk, and then you try to identify where they got the affected gene from. So complete pedigree is often a work of labor, and its finished form is frequently a work of art. So pedigree helps us a lot in genetical studies, and it also helps us analyze the risk of like the future generations, to be affected. So it gives us a lot of information about the family and also about the future. Thank you.